This isn't about inventing a new antibiotic, it's about changing how genes spread in bacterial populations. Antibiotic resistance is hard to undo because bacteria don't just evolve resistance slowly by mutation, they share it. Resistance genes move sideways between cells through horizontal gene transfer, often on plasmids. You can think of plasmids as USB drives that carry resistance genes from one bacterium to another, even across species. As long as antibiotics are around, natural selection keeps those plasmids circulating. So resistance is a population-level genetics problem, not just a problem of killing the right cells. That's where gene drives come in, or more precisely, gene drive-like systems. A gene drive is any system that biases genetic spread, so a trait increases faster than normal genetics would allow. In animals, that usually means biased inheritance through reproduction. Bacteria don't reproduce sexually, so the mechanism is different, but the idea is the same: force a genetic change to spread through a population. In bacteria, the equivalent of a gene drive uses the same highways resistance already uses: mobile DNA. Conjugative plasmids and bacteriophages already spread efficiently through bacterial communities. If resistance can spread this way, anti-resistance can too. The molecular tool that makes this possible is CRISPR. CRISPR can be programmed to recognize a specific DNA sequence, like a beta-lactamase resistance gene. When CRISPR finds that sequence, it cuts it. That cut can disable the gene, destroy the plasmid carrying it, or trigger repair that replaces the resistance gene with a non-resistant version. Now, make it drive-like. Instead of delivering CRISPR to just a few cells, you put the CRISPR system on a conjugative plasmid or inside a phage. Once introduced, that genetic package spreads on its own from cell to cell. Every new bacterium it enters gets the same instructions: find the resistance gene and cut it. There are several ways this can reverse resistance. First, plasmid curing. If CRISPR cuts an essential part of a resistance plasmid, that plasmid is lost. Without the plasmid, resistance stops spreading. Second, gene disruption. The bacterium survives, but the resistance gene no longer works, so the cell becomes susceptible again. Third, gene replacement. The resistance allele is overwritten with a susceptible version, actively editing the population back towards sensitivity. Fourth, blocking further spread. You can target genes needed for plasmid transfer, trapping resistance so it can't move. Notice that not all of these strategies kill cells. Some just re-engineer them. That matters because once resistance genes are removed, susceptible bacteria often grow faster. Even without killing cells, natural selection can now favor susceptibility again. At the population level, this flips the evolutionary logic. Today, resistance spreads epidemically because mobile DNA spreads. A gene drive-like system aims to make anti-resistance spread epidemically instead. You're not fighting evolution head-on, you're changing the rules evolution is playing by. Conceptually, this could matter in places like hospitals, biofilms on medical devices, or wastewater systems where resistance genes circulate constantly. But it's still experimental. There are serious limitations. Bacteria can evolve resistance to the drive itself through mutations or anti-CRISPR proteins. Targeting must be precise to avoid harming beneficial bacteria. Once a self-spreading system is released, controlling or recalling it is difficult. These are active research problems, not solved ones. So the core idea is simple. Antibiotic resistance spreads because genes move. Gene drive-like systems try to reverse resistance by spreading genetic tools that delete, disable, or replace those genes, using the same biological mechanisms that made resistance such a problem in the first place.